Okay, hello everybody. Welcome to the last seminar of this semester. Today we are happy to have as our guest lecturer, Dr. Serge-Yan Landau. Jan is a world-class expert in herbal nutrition and animal health. He studied from first degree to PhD in the Faculty of Agriculture at the University of Jerusalem. Since 1996, he worked as a researcher in the Volcani Institute for Agricultural Research, where he achieved the topmost research ranking full professor. During his career, Jan developed methodologies to advance knowledge in small food nutrition, in the planting and monitoring of livestock grazing under different environments and vegetation, and advanced ethno-veterinary knowledge. Supervised 17 graduate students, participated in countless international and national conferences and committees, and became the head of the Israel Small Ruminant Committee, as well as the chief editor of the international journal Small Ruminant Research. Jan received several prizes along his career, the last one in 2019. It's a Life Achievement Prize from the Israel Dairy Board. He published more than 100 articles in peer-reviewed journals and around 200 more in books, proceedings, etc. Very prolific. After his retirement from the Volcani Center, Jan joined the research group of Guy Bar-Oz as a volunteer. And since then, he's walking around the corridors, conducting research. Actually, it's a completely new topic which he's going to talk about. Relates to small ruminants, some of it. It works on fabrics that are made of animal hair that were found in various excavations. His lecture today will present his methodological development and titled Analyzing Animal Fibers in Ancient Textiles from Arid Environments through Optical Microscopy and Image Analysis. So let's welcome Jan.
Okay, so the presentation is in English with a French accent, and all this study was triggered by the amount and quality of findings at the excavations of Nahal Omer, carried out 2020-2022 by the team of Guy Bar-Oz. Nahal Omer is at the crossroads between the Haifa to Damascus or Jerusalem road and Petra to Gaza. And it's a small village encompassing about 16 houses, 1 mosque, and 6 trash mounds that are the object of this study. And we are lucky that organic matter preserves well under favorable environmental conditions, such as desiccation in hyper-arid sites like in Nahal Omer, in the Arava Desert. And it's quite easy to excavate well-preserved fabrics, like you see here. This is a cut of one of the trash middens. You can see that there are strata of darker soil. That means that they are rich in organic matter. The site was, it's a one-period site, dated on average 720 CE, and this is the period of the greatest extent of the Umayyad Caliphate, stretching from Kyrgyzstan to the Atlantic Ocean, opening many trade opportunities. And Well, why do I see that? Some pictures from, to make you understand what it is to dig in such an environment. Digging trash millets, and then sifting, picking, and finally a very big array of specimens, textiles, seeds, papyrus, basketry, feathers, ropes, dung bellets, bones, wood artifacts, human hair, a lot of it, leather, pottery sherds, and what is our subject? It is only textiles, 4,000 specimens of textiles, including vegetal fibers, cotton and linen, silk, and animal hair. At the end of this study, do somebody know how I put that off?
Yeah, you click on number.
You click on
Sorry. I did the more icon.
more. Okay. Click on more. And
hide
hide.
floating meeting control.
Here.
The last one.
One more.
One more. Yeah.
So first, looking at all these specimens, we must eliminate all the non-animal, silk is animal, but not really animal fibers. Silk is very easy to identify. You have fibroid tubuli wrapped into sericin and a groove here. You see that groove very easily on all the slides under microscope. So very easy to eliminate. We had also to eliminate linens, which is segmented with nodes and very homogeneous and fine in diameter. And cotton, which has a characteristic twisting of the fibers. And we are left with animal fiber. So what kind of animals could we find in Nahal Omer? First of all, sheep. Sheep could be, where is the sheep? Okay. We have, first of all, merino-like sheep. We'll explain more about merino, but during the Roman period, there was very tough selection towards a white sheep with white fiber. So they are all around the Mare Nostrum here, including Spain and southern France, and even to Britain, even in the Maghreb. We probably expect to meet Central Asian sheep of sheep breeds and the Levantine breed too. Here, goats, hair goats that we know from the Baladi goats here in Israel, or even Kashmir goats that thrive on the outskirts of the Umayyad Empire. And of course, we have one-humped camel, even cited in the Bible, or we have bones, and but also possibly Bactrian camels, two-humped camels that could be found in northern Persia, in Afghanistan of today, and all Central Asia, and in Mongolia, which was also a place of trade with the Umayyad. And rabbit. Why rabbit? Because rabbit for fiber was domesticated in the 7th century in Spain, and it was already the object of fabric manufacturing in Spain, but not only in Spain, also in China. I have to get rid of this one.
Right here.
The lower corner. Yeah. Okay. We can look at a sheep primitive coat. It consists of camp hairs that you see here. They are about 80 to 100 micrometers in diameter, and fine underwool of hairs that are about 10 to 20, 30 micrometers, so clearly two populations of hair. Camp hair refers to coarse fibers that do not dye well. You must remember that. And they reduce wool quality, but they can be used in carpets. And the fine fibers can be collected by plucking or combing at seasonal molting, because in primitive breeds of sheep, there was seasonal molting, and people could collect the hair by combing or even by plucking. But of course, the yield was very low. Okay. So if we consider the fleece of a wild mouflon, we can see two populations of hair. One, the underwool with very fine fibers, and then the outer coat cap. And selection of wool generally has consisted of eliminating the cap and then making the wool more homogeneous, from white mouflon to merino quality wool. And the average diameter of fibers in merino wool is about 25 micrometers. We have looked at fleeces. Now we're looking at fabrics. And we're looking at fabrics because the whole merino selection has been completed in Apulia of Italia at Roman times. Please look at fabrics found in tombs at the late Bronze Age. They still have a primitive pattern of fiber distribution. And if we look at the 4th century BC, all the camp is eliminated, possibly by genetics, but also by de-hairing, by combing the hair. And there was a revolution, which was the invention of shearing scissors. Instead of getting about 300, 500 grams of hair, you get the whole fleece, which is 2.5, 3 kilograms of hair. And of course, you get somewhat coarser wool. This is typical merino wool with a mid-fiber diameter of 23 to 27 micrometers. That's what we are going to meet at Nahal Omer. So if we consider hair materials as markers of trade routes, we can ask, does the rabbit appear in our specimens, or merino wool, or so does the rabbit appear, or merino, or Bactrian, or even do we have fiber from the cashmere goat? And that's the question of this study. We have built modern references collected from animals likely to be present in the region at the time at the Umayyad period, including sheep, goats, dromedary and Bactrian camels, and rabbits. This is the construction of a reference modern hair database with more than 300 specimens, including Awasi wool from the Negev, merino wool from the Golan Heights, camelids from one dromedary collected here in Israel at Arara in a Bedouin hut, Bactrian from the Safari and from Mongolia, goats collected from the Carmel, Ramat al-Adif, and rabbits also collected from Arara. A small collection database. We used light microscopy because, of course, there is DNA, there is proteomics, there are many methods in order to identify species in wool. But what we wanted is a cheap method with ubiquitous equipment. You have light microscope everywhere, and it should be easy transportable, and also very small samples are needed. Observations were conducted at 200 times magnification using a very good microscope, a Nikon one. The pictures was measured by using a Topica software, and you might think that it is not rocket science. I would agree with that, but the novelty here is that we have combined oil mounting, gel imprinting, and statistical models to elucidate animal species in hairs. Nobody has done that before. We look at the transversal cut of a hair shaft. We have the fiber diameter here in blue. We have the medulla, which appears as a black circle. It is full of air, but when the specimen is immersed in oil, it appears black in the microscope. We can also measure the medulla diameter. And as a matter of fact, the air spaces in the medulla provide the thermal insulating and lightweight properties in wool, which is good for the animal, but impairs dyeing, which is bad for the wool manufacturing. So if you remember, we had kemp hairs that were bad for dyeing wool, and again we have medullated hair that is also bad for manufacturing. In fact, these two populations are very similar. These are the same hairs. The proportion of fibers with the medulla is termed medulation rate. If you have 10 hairs and one of them is medulated, medulation rate is 10%. Okay. What do we see? Our program for image analysis provides, of course, the scale. It allows us to write things and notes. We can measure the distance between 2 points. Here, we can measure fiber diameter. Here also, we can measure scale length, which will reveal to be a very important estimate of wool quality and species. And all this is done with specimens that are immersed in oil and covered with a glass cover. You can see here specimens of hair that are fully medulated, like the Bactrian camel or like the rabbit. See that they are quite difficult, but different, but all medulated, or the chamois goat here, medulated. But you can also see that here the dromedary hair can be fully medulated or only by segments, like the Awasi sheep. And you will also notice that these two specimens are very similar. So, of course, it poses a statistical problem. How many? We'll deal about that. First, we look at the whole database. Medulation rate, which is the proportion of fibers with the medulla, is 12% in Avasi, 0% in Merino. You do not find a medulla in a merino fiber. And then all the others, 75, 100, 92, 100. So there is a huge difference. It is a very good discriminant of sheep wool. Medulation is a major discriminant of sheep wool. Then we can see that if we look at sheep itself, and we are looking into breeds, we see that the minimum fiber diameter is similar also the average, but we can see that in a Wasi sheep, we have gown hairs or cam hairs that are much coarser in diameter than the merino. So we will be able to define breeds within our specimens. What I wanted to show here is that even in dromedary or in goat or in rabbit, we have very, very fine fibers. But on average, they are coarse in diameter. Okay, so some statistical problems. If the incidence of medulation in sheep is 8% on average, and the unit to compute medulation rate is a slide of 5 hairs. If the population to compare sheep with has an incidence of 75% of medulation rate, then how many slides do I have to compare in order to know if it's a sheep or a camel? And in this guide, in this case, with a power of 90%, only 3 slides of sheep are needed to demonstrate that individual sheep hairs do not belong to the category of camel, goat, and rabbit with high medulation rate. But claiming that sheep as a whole differ from the rest would require 18 slides. So it's not only microscopy, it's also we must have a statistical background in order to interpret the results. If we look, this was the Awasi fiber from before. But if we look at this merino and this Awasi, we can find that this one is a merino. However, if we take 6 different slides from this wool, we see that we find medulation. Okay, so we could be mistaken by looking at too few slides of one specimen. Till now, we have done about the oil immersion preparations. Now we are going to look at imprints in gelatin. Gelatin at room temperature is solid. But if we heat it up to 60 degrees, it becomes liquid, and you can pipette it on a plate. So here we have a specimen. Here we have a drop of gelatin, liquid gelatin. We can put the specimen on the gelatin drop, wait 4 hours until it solidifies, and then we can peel the specimen off the gelatin, and we have an imprint of the cuticula. And this you will see that it is very, very useful because, for example, rabbits have very thin fibers. They have typical lanceolar cuticulae. There is no other animal with a cuticula like that. And you see here, the medulla of a rabbit is scalar. Also, you will not find medullae like that in other species. Sheep have mosaic-like structure of cuticula. And if you look at the ratio of the scale length to fiber diameter, it is 0.4. You will see that looking at camel has a very, very different cuticula with rippled, dense, concentric cuticular scale, very similar to goats. We see that they are quite similar. And the ratio of scale length to fiber diameter is 0.15. So that's a huge difference with the sheep. So in order to identify sheep, we have medulation rate. And now we have the SL to FD ratio, which is very difficult, very different from sheep from camel and goats. But everything is not that simple. For example, back-trained camel and sheep share smooth, rounded, mosaic-like cuticular scales. Here we have sheep, we have dyed it, but it's sheep. Also here, and here you see rounded cuticula. And this is back-trained camel, which looks very, very similar. But you must remember that medulation rate in camel is 100%, and in sheep only 8%. So that's not a desperate case. And finally, we have found in one camel that it has rippled concentric cuticula here and here, but also smooth mosaic-like cuticula. And of course, this is, first of all, you don't find that in the literature. And a possible explanation is that camels from the two species have been hybridized for many years in the medieval period, because hybridized camels were stronger for work, for caravans, than one-humped camels. So even in Egypt, for example, there is historical evidence from the 10th century that there were hybrids. This is the probability of a sheep, of being a sheep, using multi-binary models with fiber diameter and the probability. Please look at the mosaic structure. If you are a hair from 10 micrometers to about 100, and you have a mosaic structure of cuticula, you are a sheep. So that's so defining sheep, identifying sheep in our database is easy. Now you remember that we talked about Kashmir goats. Kashmir goats on the Pamir Plateau and in Mongolia and in etc. etc. The eastern outskirts of the Umayyad dynasty of Caliphate. This is a slide from a Baladi goat in Ramat Nadiv here on the Carmel. This is what we expect. This is exactly what we expect. All fibers are medulated. But this is another slide from the same goat, and we were very surprised to find that they have Kashmir fibers. Here in Israel, in ordinary goats. And if we look at these Kashmir fibers, there are very, very thin fiber, 19 micrometers, and they have rounded particulars. But we also see that here we have a small difference with sheep, merino sheep. It is the prominence of the scales outside of the axis. Can we differentiate once we have eliminated sheep? Can we differentiate between goats and camelids? If the fiber diameter is lower than 150, for example, it's not so easy. It's not obvious. See that? But if the fiber is more than 150 micrometers in diameter, it's a goat. There is much more probability that it's a goat. And this is the final outcome of the method. We have devised a flow chart used to differentiate between sheep, goat, and camelid hairs in the modern database to be used afterwards with the ancient hair. Less than 20% modulation is sheep or cashmere. We don't have cashmere, but we know how it looks from the literature. From 60 to 100%, so here we have a big gap. It's very, very well discriminated. Then 60 to 100% modulation, goat, dromedary, Bactrian, also rabbit, but I don't include it here. And then within each class, if we have mosaic round smooth scales or concentric smooth scales, we will have to refer to goat, cashmere, or sheep. And concentric rippled scales belong to goats and dromedary. Mosaic round to Bactrian. This is a coefficient that we have invented, the SL to FD ratio, and we can see that it's very high in sheep, 0.4. It's only 0.25 in Bactrians and 0.15 in one hemp camel and goats. And finally, in order to differentiate between goats and dromedary, we know that there is no non-modulated fiber more than 35 micrometers in diameter in goat, but we can find non-modulated hair up to more than 90 micrometers in camel. So we have a method, and now we come back to the lab where the samples are dedusted, tagged, classified, and kept at 20 degrees Celsius. And these are the results. I have included a specimen from Moa. It is a Roman toga. It is from the early Roman period in Israel. It's not manulated. We have a mosaic and annular cuticula, very small scales. Diameter of fiber is 20. The SL divided by FD ratio is 0.46. Clearly, it's pure merino wool. Don't know if the toga was moved here or it was imported as product, but it is merino. Okay, so there was merino in the southern Levant at that period. Look, please, at number 3 here. It is from Nahal Omer. We have 2 sheets of wool. One is brown and one is beige. Merino sheep are white. So here we have another breed of sheep, but still we have a fiber diameter of 24, which is very fine. Merino-like in diameter. We have a ratio of scale length to fiber diameter that is 0.4, so very cheapy. And we have modulation. So there was or here, or in Central Asia, a breed of sheep that was brown, at least partially, that felt was made of, and it's not merino, it's a local Central Asian, and it reached Nahal Onen. Here we have another felt sample made of coarser fiber. Another breed of sheep, not merino, but still white. Look at number 5. It's a beige felt. It's all modulated, concentric. But look at the fiber diameter, 70, 80, 90, 100. It's huge, very coarse. So it's not cheap. It can be either goat or dromedary or even Bactrian because of the smooth cuticle. It's probably not a goat because of the color. Most of the goat hairs were black, but it can be Bactrian. We don't know, and for that we need a DNA analysis. A very nice specimen of yarn, undyed, very merino-light, very fine. 27 is about merino quality. So another evidence of merino sheep or merino products in Israel. We had also strings. Here we have a bicolor brown and beige string, modulated, but also not modulated here. Very, very thin white fibers. So we have a very big contrast between the brown part of the string and the white one. It can be camel because we have very coarse fibers, but it can be also goat hair because of the cashmere-like fibers here. Finally, this is to show that dyeing the threads does not impair our ability to define species. We see that here we have peeled off the cuticula. We have the imprint of a thread that is dyed in green. Same here in red. So no problem. Dyeing is okay with us. And here a surprise. On the first day of analysis, we found those two. They have a scalar medulla. They have lanceolar cuticula. Obviously, they're rabbits. Same day, another rabbit in another locus. So we thought, wow, that's a discovery. We know that rabbits were used for textiles here, but alas, these were the only 2 specimens of rabbit that we found out of 300 textile specimens. So if we go to, and it's probably, here we have a hair mosaic from the area. There were hair in Israel. There were no rabbits, so no discovery. We are now working on fabrics themselves, not only materials. This is a sock that has modulated fiber and also mosaic-like. It is fine wool that is not merino. So if we want to make conclusions about what we found as to markers of trade routes, yes, there is merino wool here. There were probably merino sheep or possibly merino sheep or merinized breeds of sheep, like all around the Mediterranean. We are less confident that there are Bactrian, even though the fibers that we found could fit very well to Bactrian camels, or at least to hybrids. So Merino-like and Levantine or Central Asian sheep, local goats, dromedary, and probably Bactrian were here at that period. We don't think that rabbit was an issue here, and there was no evidence of cashmere, of pure cashmere. If we look at the Nahal Omer assemblage, it was 68% of it was consisted of sheep, 17% of camels, 4% Bactrian, 2 Hapt, 10% goat, and the rest rabbit. But if we look at the different categories, you can see that, for example, in strings, we have 85% of goat and camel. We look at yarn, we have almost 100% of sheep. And we reach the conclusions. The level of conservation of ancient textile materials in Nahal Omer was good enough to assess animal hair species by light microscopy at high confidence level. Combining oil preparation and gelatin imprinting enables the identification of sheep hair in fabrics at about 100% of confidence. Sheep wool represented 68% of hair materials. The distinction between dromedary camels and goats is satisfactory only for coarse hair. Dromedary represented 70% of hair materials. Goat formed about 10% of Nahal Omer materials. Probable evidence of bacterial movement. Rabbit's hair had unequivocal medulla and cuticular attributes. Rabbit was probably not an important component of the Nahal Omer textile materials. I would say it was not a component. And what next? The database, the variety of the assemblage in Nahal Omer is huge. So first, we are going to complete wool and fabrics from Nahal Omer analysis, like the one you've seen now, until June, I think. Then we would like to do DNA to define wool provenance, in particular to distinguish between Bactrian and dromedary camels, because it tracks trade routes, animal breeds, and hybridization also. Ancient DNA in wool is not very easy, but it's feasible. So I hope that we can do it. We would like to analyze pigments, dyes, and inks, chemistry, and provenance. That would also shed light on trade routes. We would like to differentiate merino and cashmere hairs by scent, because it's the only possibility to do it. And of course, we have leather and accompanying hair. We have there is a lot of this, and we must learn about species and provenance. And there is tons of human hair of different colors and with different curvatures, and which could reveal a lot about human activity in the site. And that's finished for today. I must thank you, Guy, for hosting me as a volunteer in your lab. No far from managing my time very well and efficiently. Ruth and Gal for tutoring us in microscopy. Itza for the statistical models that you haven't seen here, but are quite impressive. Harry for doing the hard work with the microscope and doing it well. And of course, we also thank Roy for getting bacterial hair. He had to fill a lot of paperwork and with the safari, and that's quite incredible how just to clip a few hairs. Gidi also that provided hair from Gulia. Raid Mahdi and Michal from the Ministry of Agriculture, who provided us with hair from the Negev and Ramat HaGolan and the Golan Heights, and Najjar Halabi from the Ramat HaNanev, who provided the goat hair. And this is very nice to conclude. It is stamp, an ink stamp on one of the samples. And we believe that by analyzing the composition, we have found in the literature similar stamps from Turkmenistan. But we must be sure, and so thank you for your attention. Yes, Mary.
Thank you, Jan. Very novel from someone that comes from the animal aspect to hear another side. My question is related actually to cattle. Are you able to find any evidence that also you found the hair of cattle also in this bulk of hair that can imply that cattle was also in the
Cattle. We have cattle in our collection or in our database. There was no cattle. You must understand that it is not. This is trash minerals. So this we collect only. Maybe there was cattle. Maybe there was cattle. We know how cattle must look like. But if any cattle was not imported, we didn't find one in 288. And also, we know that sheep was the preferred animal there. Cattle was, if any, very small in frequency in the Negev. Yeah.
Much young. My favorite is 100% Kashmir. But when you read the labels, you often see 90%, 10% Kashmir. You know, a combination of lightness, strength, etc. My question to you is whether through the natural assemblage or maybe literature, you know if it's customary in the textile industries to combine more than a single raw material in a piece of fabric.
Yeah, of course, it's also illegal. Yeah, because there are now, well, even though it can be labeled, so but we have many evidence, much evidence that that Kashmir is mixed and sold under the Kashmir tagging. And there are, for example, there is this the control of Kashmir is done also under microscope. They have much more performing method that we have that can proceed thousands of fibers a day, and that and you can get some idea of the authentication of Kashmir out of the textiles. Yeah, but yes, it is difficult to distinguish between Kashmir wool and very fine merino wool. Very difficult.
Much for this lecture. I was wondering, you said that the Romans preferred white wool, and I was wondering if the findings you find in Nahr al somehow correspond to what is known about fabrics in the Umayyad.
About?
To preferences in Umayyad fashion or Umayyad trading locations.
You speak about the color.
Well,
What was the preference?
Also animal hair, also
The preference, the high prices, and so on, have always been for white fiber. And you know, for example, there was merino wool in Spain at the early medieval periods, but it was not. They were not allowed to export it anywhere because they didn't want the competition. So white, fine, unmediated wool is the preferred one. But for garments and so that here you have caps, and you have shoes, and you have many other things that are made of wool, but not fine wool. They can be made of felt. For felt, you don't need very fine wool. So you could say that for fine, very fine textiles, merino is the most appreciated. But for the rest, for example, tents were in Central Asia were made of felt, but not of no fine wool. They were. So that's the preference. Yeah. Yeah.
Is it ever like you feel like it's not really very
Yes. There is a very nice paper by Gutwein that shows how ships were looking at the Byzantine period according to mosaics. You see, when you have rolling hair, it is a sign of, it is an icon of merino, and when you don't have that, it is coarser hair. And for example, you know that the merino have no fat tail, but here in the Levant, a very fine fiber sheep wool. Sheep they were merino in wool quality, but they had a fat tail, very long fat tail, not like the Abassi of today, but they had it. Yeah.
Ask a technical question. How long did it take you to develop the method? Also, did you start the project thinking that you're going to develop a method, or it came something along the way?
No, I did it. I came to a guy to volunteer, and he told me, "Okay, you're a consultant on olive trees." What was it? Palm trees. But there was nothing to do there, so go to Nofar. She has to dedust specimens. It was in January 24, and we had to collect everything. In 1 year, we had the method.
In 1 year.
2 days a week.
Very fast. Right.
Building the database
Yeah.
using modern today here.
Yeah.
First, did you take into consideration the probable differences between hair taken at a certain season
Yeah.
of the year and same hair from the same animal taken at a different season? And second, how do you overcome the probability of hybridization in animals that we have today that took place during the time between specimens that you found were created and today's hair that you use as a database?
Well, first of all, merino is merino. You cannot be mistaken. I mean, it was merino at the early medieval period, and you have literature, and also all the people have worked about that. They had no microscopes, but fiber length and curvature and that kind of things. And our merino in Ramat Golan, in the Golan Heights, you didn't find even one medulated specimen. So I don't think that they were different from those in there are lesser. Of course, there is maybe selection for all the attributes that are meat quality and that kind of things, but I don't think that they're very different. Then when we constructed the database, we used places, not animals. We went to Arara, and there were huge stacks of shorn, of wool that had been shorn a few days ago, and we selected from inside the sacks. We didn't have an animal bias. It was random, but it is at the period of shearing. So we don't have, you make wool in the spring. The composition of the fleece in the autumn, when the animal are not shorn, is not important in that point, and it was always that same management. So, but you're right that you can have differences between different anatomical points of an animal, but here we didn't. But we used individual animals, only in camels and bactrians, because we had no possibility to do otherwise. But for the others, there is no bias.
Once again, yeah.
So, so one thing is to remind us all that next week there is no lesson. Remember this seminar next week. Meet again in 2 weeks' time for our last meeting, and then we will discuss this presentation that we saw today. We will discuss the article. Yes. Okay. And we'll have a guest performance by Mufal Shamil, was also in the presentation. She was working on the textiles under conservation and preservation. So we're going to talk a little bit also about conservation and preservation of these very, very delicate artifacts and how that goes. So she will also maybe send us another article mostly on that subject for you. Okay.
Have any general impressions about
Just for us, something about the presentation. Right, the dyeing doesn't really change the structure.
said, so we are checking. Is there anybody expert on this? That's not. We have any experts.
Very interesting that you have to make this.
Developing a method in any field is really not easy. Actually, developing this so fast in 2 days' work per week for 1 year is really remarkable. The method can be applied to archaeological materials. The patients start to flow in. First, I think that you can apply to a specific research question, and other people are going to read the article and have ideas and apply to other research questions. And such methods like what you have developed has the potential to become an international database. It's going to increase research. Some people are excited about methodology. Yeah. So if there are no more comments or questions, we're going into it. Remember that